evolution does not have a chance to explain how the blood coagulating system works. Michael Behe again. Is it possible that the ultra-complex system could have evolved according to Darwinian theory or neo-evolution and all the other forms of evolution? The observation of split genes where similarities were observed in the genetic structures of different parts of a biological systems led to the hypothesis that perhaps new proteins could be made by shuffling DNA fragments of genes that code for parts of old proteins, much as cards can be picked from several piles to give a new arrangement. To support the hypothesis, advocates point to similarities in amino acid sequences and shapes of discrete portions called domains of different proteins. I've heard this before. Just doctrine of details. Get into details. The proteins of the blood coagulation cascade are often used as evidence for shuffling. Some regions of cascade proteins coded by separate gene pieces have similarities in their amino acid sequences with other regions of the same protein. That is, they are self-similar. Also, there are similarities between regions of different proteins of the cascade. For example, proconvertin, Christmas factor, Stuart factor, and prothrombin all have a roughly similar region of their amino acid sequences. Additionally, in all these proteins, the sequence is modified by vitamin K. Furthermore, the regions are similar in sequence to other proteins not involved with blood coagulation at all that are also modified by vitamin K. The sequence similarities are there for all to see and cannot be disputed. By itself, however, the hypothesis of gene duplication and shuffling says nothing about how any particular protein or protein system was first produced, whether slowly or suddenly, or whether by natural selection or some other mechanism. Remember, a mousetrap spring might in some way resemble a clock spring, and a crowbar might resemble a mousetrap hammer, but the similarities say nothing about how a mousetrap is produced. In order to claim that system developed gradually by a Darwinian mechanism, a person must show that the function of the system could have been formed by numerous successive slight modifications within an acceptable time frame. By the way, you can't say given enough time because if the universe is not long enough in its existence, according to evolutionists, then the blood clot system is not feasible. The first thing to notice is that no causative factors in the evolutionary models proposed are cited. Thus, tissue factor appears, fibrinogen is born, antiplasmin arises, TPA springs forth, and a cross-linking protein is unleashed, and so forth. What exactly, we might ask, is causing all this springing and unleashing? Step-by-step Darwinian scenario involved the undirected random duplication and recombination of gene pieces is in view. A step-by-step Darwinian scenario involving the undirected random duplication and recombination of gene pieces is in view when you consider if evolution could do this. But consider the enormous amount of luck needed to get the right gene pieces in the right places at the right time, often simultaneously, or precise sequentially. Eukaryotic, organisms have quite a few gene pieces, and apparently the process that switches them is random. So making a new blood coagulation protein by shuffling is like picking a dozen sentences randomly from an encyclopedia in the hope of making a coherent paragraph. Furthermore, the Darwin scenario typically does not go to the trouble of calculating how many incorrect, inactive, useless, varying shuffled domains would have to be discarded before obtaining a protein with, say, TPA-like activity. To illustrate the problem, let's do our own quick calculation. Consider that animals with blood clotting cascades have roughly 10,000 genes, each of which is divided into an average of 3 pieces. Thus, this gives a total of about 30,000 gene pieces. TPA has 4 different kinds or types of domains, and by various shuffling, the odds of getting those four domains together is 30,000 to the fourth power, which is approximately 1/10 to the 18th power. Now, if the Irish sweepstakes had odds of winning of 1/10 to the 18th power, and if 1 million people played the lottery each year, it would take an average of about 1,000 billion years before anything, not just a particular person, won the lottery. 1,000 billion years is roughly 100 times the current estimate age of the universe. The same problem of ultra-slim odds would trouble the appearance of prothrombin, fibrinogen, plasminogen, proaccelerant, and each of the other several proposed rearrangements of prothrombin. Unfortunately, the universe doesn't have time to wait. The second question to consider is the implicit assumption that a protein made from a duplicated gene would immediately have the new necessary properties. Thus, we are told by the Darwinian model type, tissue factor appears as the result of the duplication of a gene for another protein. But tissue factor would certainly not appear as the result of the duplication the other protein would. If a factory for making bicycles were duplicated, it would make bicycles, not motorcycles. That's what is meant by the word duplication. A gene for a protein might be duplicated by a random mutation, but it just does not happen. It does not just happen to also have sophisticated new properties. Since a duplicated gene is simply a copy of the old gene, an explanation for the appearance of tissue factor must include the putative route it took to acquire a new function. This problem is discreetly avoided. The Darwinian model runs into the same problem in the production of prothrombin, a thrombin receptor, antithrombin, plasminogen, antiplasmin, proaccelerant, Stuart factor, proconvertin, Christmas factor, antihemophiliac factor, and protein C, virtually every protein of the system. All together simultaneously, no, it's not going to happen. The third problem in the blood coagulation scenario is that it avoids the crucial issues of how much, how fast, when, and where. Nothing is said about the amount of clotting material initially available, the strength of the clot that would be formed by a primitive system, the length of time the clot would take to form once a cut occurred, what fluid pressure the clot would resist, how detrimental the formation of inappropriate clots would be, or 100 other such questions. The absolute and relative values of these factors and others could make any particular hypothetical system either possible or much more likely wildly wrong. For example, if only a small amount of fibrinogen were available, it would not cover a wound. If a primitive fibrin formed a random blob instead of a meshwork, it would be unlikely to stop blood flow. If the initial action of antithrombin were too fast, the initial action of thrombin are too slow, or the original Stuart factor or Christmas factor or antihemophiliac factor were bound too closely or too tightly, or if they bound to the inactive forms of their targets, as well as the active forms, then the whole system would crash. At no step, not even one, does any of the Darwinian models give a model that includes numbers or quantities. Without numbers, there is no science. When a merely verbal picture is painted of the development of such a complex system, there is absolutely no way to know it if it would actually work. When such crucial questions are ignored, we leave science. Yet the objections raised so far are not the most serious. The most serious, and perhaps the most obvious, concerns irreducible complexity. I emphasize that natural selection, the engine of Darwinian evolution, only works if there is something to select, something that is useful right now, not in the future. Suppose, for purposes of discussion, as might occur in a typical Darwinian model, no blood clotting appears until at least the third step. The formation of tissue factor at the first step is thus unexplained, since it would then be sitting around with nothing to do. At the next step, prothrombin popping up already endowed with the ability to bind tissue factor, which somehow activates it, the poor prothrombin would also be twiddling its thumbs with nothing to do until at last a hypothetical thrombin receptor appears at the third step, and fibrinogen falls from heaven at step 4. Plasminogen appears in one step, but its activator TPA, doesn't appear until 2 steps later. Stuart factor is introduced in one step, but whiles away its time, doing nothing until its activator, proconvertin, appears in the next step, and somehow tissue factor decides that this is the complex it wants to bind. Virtually every step of the suggested pathway faces similar problems. It goes on and on and on. Simple words like the activator doesn't appear until 2 steps later may not seem impressive until you ponder the implications. Since 2 proteins, the proenzyme and its activator, are both required for 1 step in a pathway, then the odds of getting both the proteins together at roughly the square of the odds of getting 1 protein. Unbelievably long time. We calculated the odds of getting TPA alone to be 1/10 to the 18th power. The odds of getting TPA and its activator together would be about 1/10 to the 36th power. That is a horrendously large number, since an event would not be expected to happen even if the universe's 10 billion year life were compressed into a single second and we lived every second for 10 billion years. But the situation is actually much worse. If a protein appeared in one step with nothing to do, then mutation and natural selection would tend to eliminate it. Since it is doing nothing critical, its loss would not be detrimental, and production of the gene and protein would cost energy that other animals aren't spending. So producing the useless protein would, at least to some marginal degree, be detrimental. Darwin's mechanism of natural selection would actually hinder the formation of irreducibly complex systems such as the Clotting cascade. Unbelievably complicated. The bottom line is that clusters of proteins have to be inserted all at once into the cascade. This can be done only by the guidance of what? An intelligent agent. A little bit more than ape man, right?